and welcome to this class. I am Ngubani Kestin and I'll be taking on the subject chemistry. The theme of our class is chemistry of life and our topic is proteins. At the end of our lesson, you should be able to list the sources of proteins and give examples. You should also be able to identify amino acids as the building blocks of proteins. You should be able to state the physical and chemical properties of proteins, carry out tests for proteins, and you should be able to state the uses of proteins. Now, proteins are found throughout the body. They are in your hair, in your nails, in the muscles and skin. As humans, our bodies are composed of about 10,000 different proteins, which account for about 20% of our total body weight. Yes. Proteins make up enzymes in our bodies and in other living organisms, hormones, neurotransmitters, and tiny molecules that power many chemical reactions in the body. This is why they are called the building blocks of life. In fact, without proteins, life as we know it would not be possible. Now, let us take a look at what proteins are. So that proteins are molecules that are complex, and they are made up of chemical compounds called amino acids. So they are polymers of amino acids joined together by peptide bonds. So it means that amino acids are actually what come together to make up proteins, which is a complex structure. Now, here we have an image, of a typical protein. It's a complex structure. As you can see, each of these circles represents different amino acids making up this total complex structure called protein. Now, amino acids are linked together like beads on a string. Looking at this diagram, it looks like a bead, right? So they are linked together like beads on a string, forming long protein chains, which can now be folded into more complex shapes. Now, amino acids are known as the building blocks of proteins. Just as a child uses these tiny, beautiful, and colorful boxes to produce a giant playhouse, amino acids can be likened to these smaller boxes that are now being piled up individually to form a very big protein structure. So when we say amino acids are building blocks of proteins, you can actually liken them to building blocks. So they make up what we call the protein structure. Now we'll be looking at amino acids as the building blocks of proteins. We say that amino acids are organic molecules made of carbon, hydrogen, nitrogen, oxygen, and sometimes sulfur. They contain a basic amino group and an acidic carboxyl group. Here we have this diagram of an amino acid. You can see the amino terminal and we can see the carboxyl end. Every amino acid must have an amino terminal, also called an N-terminus, and a carboxyl end, known as C-terminus. Now, in proteins, what happens is that the amino acids are linked together. An amino acid will be linked to another, which will also be linked to another, and when they are built up like that, they form proteins. The bonds that link one amino acid to another is what we call a peptide bond. Now, when two amino acids join together, the bond between them is called what? A dipeptide bond, signifying that it's just two amino acids. When three are linked together, their bonds are called tripeptide bond. And when multiple amino acids are linked together, the bonds are called polypeptide bonds, but most times we just call the bonds peptide bonds for simplicity. Now, there are about 22 different amino acids that basically make up proteins on Earth. The amino acids are listed here. We have alanine, cysteine, aspartic acid, glutamic acid, phenylalanine, glycine, histidine, isoleucine, lysine, leucine, methionine, and the rest. Now, out of these 22 amino acids, 9 of them are called essential amino acids, while the rest are known as non-essential amino acids. The ones called essential amino acids are the ones we have here in this box. And they are called essential because they cannot be made by the body. So we need to obtain them from food sources. Okay? While the other kinds of amino acids can be synthesized by the body. And so they are called non-essential. The 9 essential amino acids are leucine, isoleucine, valine, histidine, lysine, methionine, phenylalanine, threonine, and tryptophan. We have to obtain these amino acids from the food we eat. Okay? Now remember that these amino acids, whether the essential ones or the non-essential ones, are very, very important for our bodies. Okay? For protein to be synthesized in our bodies and for our body to function properly. So we must obtain these essential amino acids from the food we eat so that they, in combination with the non-essential amino acids, can be used to synthesize proteins in our bodies. Now, these amino acids, these 22 amino acids, can be arranged in millions of different ways to obtain different kinds of proteins, millions of proteins, okay? And each protein has a specific function in the body. Remember, we said that enzymes are proteins, hormones are proteins. There are lots of neurotransmitters in the bodies that are proteins. So they are at one point or the other in our bodies carrying out different specific functions. Now, we have said that amino acids can be arranged in millions of different ways. For example, we can see in this diagram, we have different proteins. What makes them different is that there are different sets of amino acids that make them up. This first protein have a different set of amino acids from this second protein, also this and this. So amino acids can be arranged in millions of different ways to obtain different types of proteins. Now, each amino acid must have a central carbon, okay, usually called the alpha carbon, bonded to a hydrogen atom. So this is a typical amino acid structure. It must have a central carbon. This carbon is what we call the alpha carbon. And it must have a hydrogen atom bonded to it. Now we'll be looking at different sources of proteins. We have plant sources and animal sources of proteins. Plant sources of proteins include vegetable food items such as legumes and nuts. Now, plant sources of proteins do not provide the body with all the essential amino acid it needs. Okay? Therefore, plant sources of proteins are considered incomplete sources of proteins. Animal sources of proteins include meat, for example, cattle, fish and seafood, and also poultry. Animal proteins are considered complete source of protein because they provide the body with all the essential amino acids it needs to function effectively. Now, there are 4 protein structures, okay? And they are arranged in levels based on the 3-dimensional arrangement of atoms of the protein molecule, okay? Now, these levels go from the first level, which is the primary structure, and then it falls into the secondary structure, and then the third level, which is tertiary structure, and the final level, which is the quaternary structure of proteins. We'll take a look at each of these structures. Now, for the primary structure, this is the first level, okay? For proteins to be formed, they first begin as straight chain or sequence of amino acids. So the first time that amino acids start building up makes up the primary structure of proteins. For example, we can see this protein is just a linear structure of amino acids that are held together by peptide bonds. There are peptide bonds between each of these amino acids. So an example of protein with primary structure is the insulin. It is composed of 51 amino acids present in 2 chains. This is the first chain, this is the second chain. This is an example of a protein with the primary structure. Now, going over to the secondary structure of amino acids is the second level. What happens here is that beyond the primary level or straight chain of amino acids, okay, each of these amino acids begin to interact with others to form a regular pattern. And this formation is due to the hydrogen bond between them. So the amino acid chains, which are the polypeptide chains, begin to twist and coil, creating a 3-dimensional geometry like an alpha helix, like what we have here in this diagram, or a beta sheet, looking like this. So this is the secondary structure of a protein, okay? It moves from primary level to the secondary level. The primary level, we just have a linear or straight chain of amino acids. In the secondary level, these amino acids begin to interact with each other and begin to coil, fold, twist, to form this secondary structure. And this is made possible by hydrogen bonding between them. And this folding is in a regular pattern, and they form what we call either alpha helixes or beta sheets. Now, there are some kind of interactions that, help these proteins to fold in a certain way. We have hydrophobic and hydrophilic interactions and hydrogen bonding, okay? Hydrophobic would mean parts of the protein that are water repelling, okay, that do not like water. So they tend to fold inwards or fold away from water, while the hydrophilic ends or parts of the protein tend to interact with the water. So these hydrophobic and hydrophilic interactions, coupled with the hydrogen bonding that go on between each of these amino acids, play a part in the twisting and coiling of the amino acids. Myoglobin, okay, is an example of a protein with secondary structure. Now moving to the third level, which is the tertiary structure of proteins. This level describes the overall shape of the protein. So what happens is that the alpha helix or beta sheet of the secondary structure, remember, keeps folding or twisting until it achieves a true 3-dimensional structure of the polypeptide chain. A tertiary structure would look like this, okay? So from the secondary structure, remember, we notice some coiling and twisting in the secondary structure. But in tertiary structure now, there is more coiling and folding or bending of these amino acids, resulting in something like this. Now, the polypeptide chains fold in a way that the non-polar or hydrophobic amino acids point inwards, okay? So the parts that are actually convoluted inwards are the hydrophobic amino acids. They tend to shy away from water, while the polar or hydrophilic amino acids point outwards, okay, where they interact with the water, resulting in the huge folding of the protein structure.